Welcome to the last day of Kidney Week 2020 Reimagined. Thank you all for joining us on this digital meeting journey. Please remember that all the meeting content will be available on this website through December 4th, including the plenary presentations. Today's state-of-the-art lecture will be delivered by Joan Steitz on RNA biology and medicine, a journey of unexpected discovery. Dr. Steitz is Sterling Professor of Molecular Biophysics and Biochemistry at Yale University and an investigator at the Howard Hughes Medical Institute. Dr. Steitz was publishing significant findings as early as 1967, when her doctoral thesis with mentor James Watson examined the test tube assembly of an RNA bacteriophage known as R17. She spent the next 3 years in postdoctoral studies at the Medical Research Council Laboratory of Molecular Biology in Cambridge, England, where she used early methods of determining the biochemical sequence of RNA to study how ribosomes know where to initiate protein synthesis on bacterial mRNAs. In 1970, she was appointed Assistant Professor of Molecular Biophysics and Biochemistry at Yale, becoming full professor in 1978. She established a laboratory at Yale dedicated to the study of RNA structure and function. Perhaps her biggest breakthrough came in 1979, when Dr. Steitz and her colleagues described a group of cellular particles called small nuclear ribonucleoproteins, a breakthrough in understanding how RNA is spliced. Subsequently, her laboratory defined the structure and functions of other non-coding RNPs, such as those that guide the modification of ribosomal RNAs and microRNAs, as well as several produced by transforming herpes viruses. Dr. Steitz has mentored over 200 young scientists. She has advocated for inclusion of more women in the sciences, and she co-authored a 2007 report for the National Academy of Sciences outlining barriers to the participation of women in sciences and steps to address those barriers. In 2018, she received the Lasker Koshland Award for Special Achievement in Medical Sciences for 4 decades of leadership in biomedical sciences, exemplified by pioneering discoveries in RNA biology, generous mentorship of budding scientists, and vigorous and passionate support of women in science. Among her many other honors are the U.S. Steele Foundation Award in Molecular Biology, National Medal of Science, Federation of American Societies for Experimental Biology Excellence in Science Award, RNA Society Lifetime Achievement Award, Gardner Foundation International Award, Perlmeister Greengard Prize, the Grand Medal from the French Academy of Sciences, American Society of Biochemistry and Molecular Biology, Herbert Tabor Award, American Chemical Society Biopolymers, Murray Goodman Memorial Prize, and the Jonathan Kraft Prize for Excellence in Cancer Research. Now, please turn your attention to a researcher who has devoted her career to investigating RNA.
So good morning, everybody. And thank you very much, Linda, for that lovely introduction. When Peter Aronson called me up or contacted me and asked whether I would give this presentation, I said, well, what do you want me to talk about? And he said, well, it should be somewhat autobiographical, and you should talk about women in science, but you should also be talking about your science, and which seems a lot for 25 minutes, but he made up this title for me, and I think it's a perfectly lovely title. So what I'm specifically going to talk about is, first I want to talk about how I got into science and particularly into RNA biology and ended up at Yale, and then a bit about the science and its impact both from medicine and on medicine. And then in the end, I'll have a few minutes about reflections on women in science in general. So to go back to the beginning, I grew up in Minneapolis, and I happened to be fortunate to go to a small liberal arts college in Ohio named Antioch College. And what was unusual about it was it had a work-study program where students didn't spend full time on campus doing book learning, but rather were sent off to various parts of the country and the world to do real jobs. This was the beginning of molecular biology, and I was very fortunate to be assigned in a job in Alex Rich's lab at MIT. Rich was one of the first molecular biologists. And the time was not quite 10 years since the discovery of the double helix of DNA, but it was still too new to have made it either into textbooks or even into lectures. But when I arrived at MIT and learned about this, I was absolutely enthralled because I thought this double-stranded structure can explain so much about heredity, which had fascinated me as a high school student. And I was clearly hooked on the subject. Nonetheless, I decided that I should apply to medical school. And that was because when I looked around me, I never saw a female professor of science or head of lab, but I did know several women physicians. That all changed just the summer before I was about to go off to Harvard Medical School, when I ended up working in Joe Gall's lab at the University of Minnesota. He was later at Yale, a very famous cell biologist who you may know and discovered in situ hybridization. And he gave me my own project for the first time. And again, I was so excited. By August 1st, I decided, no, I want to go to graduate school and make discoveries rather than go to medical school. And I was able to arrange, with his help, switching to the graduate program at Harvard. The way I got to Jim Watson's lab was that after a year of courses at Harvard, I had to choose a lab to work in. I wanted to do something similar to what I'd been doing with Joe Gall. And I went along to a very famous Harvard professor and said, I'd like to work in your lab. And he took one look at me and he said, but you're a woman and you'll get married and you'll have kids. And what good will a PhD do you then? I managed to make it out of his office before I burst into tears and went along to my second choice advisor, who was Jim Watson and Crick. And Jim and his lab were absolutely wonderful. It was the best thing that ever happened to me. By the time it was time for a postdoc, I had married my husband, who was an X-ray crystallographer. So we had to go to the mecca of X-ray crystallography in the world, which is the MRC lab in Cambridge, England. And that's how I got there. So let me show you some few pictures from this very early stage. So here's a picture that occurred at about that time on the cover of Newsweek with Alex Rich on the left and John Warner, who was the graduate student who was in charge of me on the right. And John Warner was the first person who introduced me to RNA and to research projects having to do with RNA. The next slide is a picture of the Watson group after I joined it around about 1965. I'm up on the top, second to left with my mouth open. And in the picture in the bottom right is Jim Watson, who was a recent Nobel Prize winner at that time. And next to him, Wally Gilbert and a couple people away, Mario Capecchi, who were future Nobel Prize winners. And it was an absolutely wonderful group with all sorts of various types of people in it. And here's a picture of my husband and I as we were about to take off for our postdocs in Cambridge, England. So now I'd like to go back to my outline. And at this point, I want to start talking about science. And I'll talk about how we got to Yale first from Cambridge, England, because that is pretty much a science story. And then I'll be telling you about the sort of crux of what I want to discuss with you, which is an interesting story because it's one where tools provided by clinicians, in fact, have been very, very important in unraveling the basic molecular biology of cells, sort of backwards of what we think of as bench to bedside. So what I will talk about, not talk about, is shown here. The first item is what I did as a postdoc, which actually got me the job at Yale. The second is the use of autoantibodies from patients with lupus that I just mentioned, and the discovery of the splicing SNRPs, which is a bedside to bench story. But then at the end, I want to include a little bit about how it's gone back to prove useful, very useful in medicine. I won't talk about the rest of the bullet points here, but there are lots of other things that we've gone on to do over the years. And right now I'm very excited about the last item where we're actually looking at RNA structures and seeing quite novel things about the interaction of poly-A tails on messages with the rest of the messenger RNA and how those act to stabilize those messenger RNAs inside cells. So when I got to the MRC lab, I had a spot, but I wasn't told what to do. I was told to go figure out what I should do. So I went around and talked to everybody in the lab, in a whole division, actually, who were mostly other postdocs from the US, all male, of course. And they all said, there's this really exciting project that somebody should do, which is to bind ribosomes to a phage messenger RNA that has 3 genes on it, use the binding to protect the parts of the messenger RNA then that are the beginnings of where ribosomes start translation, and then pull out those fragments and sequence them by the new methods that Fred Sanger had just developed at the MRC lab. The problem was that none of the men wanted to take this project on because they knew that they'd have to get a job back in the States in 2 years. But for me, I never expected to have a job as an academic job. So I said, gee, that sounds like an exciting project. Why don't I do it? And I did it and it worked. And it was very exciting because it was the first glimpse we had into where on a polycistronic messenger RNA ribosomes actually bind and start. And it was something that we then continued to work on, as is shown in the bottom of the slide, after I came to Yale. And we were able to show that there, in fact, was base pairing between the messenger RNA and the ribosomal RNA. And up at the top, I call it single molecule ribosome profiling because it's done with actually just one messenger RNA rather than what's done nowadays with ribosome profiling. In order to tell you about how I got from translation to lupus and SNRPs, I need to go back to the early 1970s in molecular biology. And at the time, what had been worked out by the field was the broad outlines of DNA synthesis, how RNA is made, protein synthesis. And actually, the field was pretty smug in the early 1970s. It thought, well, gee, we figured out all the major features of how gene expression works. The golden age of molecular biology is over because when we move on to higher organisms, there's not going to be anything new, just a little bit more complication. But what's shown in this slide is the one sort of fly in the ointment at the time, which was that if one looked at the amount of DNA in a bacteriophage on the left with about a couple hundred genes, a bacterium in the middle with 10 times as much DNA and about 10 times as many genes, or a mammalian cell, which has 1,000 times more DNA. And nobody thought that mammalian cells would have 1,000 times more genes than bacteria. So what was all this extra DNA doing? And also, what was known was that there was a lot of RNA synthesis in the nucleus of mammalian cells, but that most of that RNA just got degraded and only about 10% of it made it out to the cytoplasm as messenger RNA. So there was something very funny going on there. And when I had my first sabbatical from Yale in 1976-77, I decided I'd try to work on this problem. And what I wanted to do was shown in this slide. What we're looking at is DNA with RNA strands coming off of it, and then being decorating those RNA strands are abundant nuclear proteins that coat the newly synthesized pre-messenger RNAs. And I thought, well, maybe those proteins have something to do with the decision about which RNAs get degraded and which RNAs make it out to the cytoplasm. And I thought if I had antibodies against those proteins, they'd be really, really useful in studying this problem. So I tried for 7 months to make antibodies against these proteins isolated from rat liver. Nothing ever showed up, primarily because these are very highly conserved proteins. And I gave up and did something else for the rest of my sabbatical. However, as you all know, in 1977 was the year when interruptions in eukaryotic genes were discovered in the form of introns separating exons, a good part. And it then became apparent that this was all done on the mRNA level and that the message was then spliced back together somehow in cells. And Sharp and Roberts received the Nobel Prize for that significantly later. This, however, moved the molecular problem just sort of one step ahead, the question being, what is there at the junctions between the sense regions and the non-sense regions that are the underlying regions here that tell the cellular machinery this is a boundary and you have to make very precise cuts at those boundaries and then join back together the good regions in order to make a functional messenger RNA. So when I came back to my lab in the fall of 1977, everybody wanted to work on this problem because it was intriguing and we were an RNA lab, but quite frankly, we were sort of clueless as to how to begin. But then what occurred was an important piece of serendipity. And you'll see serendipity enters in several places in my discussion. What happened was that at the end of January 1978, a new article appeared in the journal Nature, and it had this introductory paragraph where I've underlined a salient sentence that says, patients with MCTD have high titers of antibody to nuclear ribonucleoprotein. And the reason that caught my eye was while I was trying to make antibodies and failing, several people had said to me, you know, I think I've heard that there are patients that make antibodies against things in the nucleus that are RNA and protein, and maybe those are what you're looking for. But at that time, I didn't know who to ask. However, when this article came, I had a brand new MD PhD student in my lab, Michael Lerner. And I said to Michael, do you know anybody here at Yale that might have patients like this that we could see about getting some serum from and see if they have antibodies in them? And Michael said, sure, I'll go see Hardin. Hardin turned out to be in the rheumatology section, John Hardin, just across the street from us in a different building in the Department of Medicine. And that very afternoon, Michael came back with a few little vials of serum and started to work with them. Now, the sobering part of this story, as you're all thinking, is that, of course, nowadays, one would have to spend weeks, months, perhaps years filling out human investigation forms before you could even use a few mils of serum that somebody would willingly give up for research. But that wasn't the case back in 1978, so Michael proceeded. And the idea was, as we found out from reading the literature a little bit, patients whose serum he had obtained were known by the clinicians to contain autoantibodies against their own cellular components that then can form immune complexes with their target antigens. And as you all know, probably better than I, this accounts for a large fraction of the pathologies in lupus and the related rheumatic diseases. But it can also be used as a tool to try to isolate the cellular component that's the target of the autoantibody. And this is what Michael tried to do. But the going was very rough really because we didn't have modern methods for detecting antigen-antibody complexes or immune complexes. And then another piece of serendipity happened, which is that a young postdoc by the name of Joan Brugi, some of you may know, has just finished many, many years as a very successful chair of the cell biology department at Harvard Medical School, came to Yale to give a talk and talked about her postdoctoral work that involved using a new reagent called pansorbin, which is really just formalin fixed Staphylococcus aureus cell walls, which you will know has protein A in them, and that protein A interacts with the Fc portions of antibody molecules and can be used to pull out complexes. So what Michael did at that point was to take HeLa cells, label them with P32 so that you label all the RNA molecules. And on the left-hand side, you see in the gel RNA molecules ranging in size from about 75 on up to about 200. When mixed with Michael's own serum, the next lane, normal serum, happily, he doesn't have any autoantibodies, so you see nothing. But if you mix it with sera from the various patients obtained from John Harden, you can see various subpatterns. And the one we're going to be concerned with here is the one in the third lane called anti-SM, where the actual antigen are proteins that are common to several different RNA protein particles. The first top two bands are U2 and U1. They'd been described in the literature, but U4, U5, U6, going down to the bottom, were new, and so we named them and characterized them. And then we also had sera that just seemed to recognize the particle that had U1 in them. By doing similar experiments using labeling with S35 methionine instead of phosphate, we could label the proteins. And from this, we realized that some of the proteins were in common between the various RNA-containing particles, but that some of the proteins were also unique, and those are the ones with the green or the blue designations in this slide. We also realized at that point that the sequence at the 5' end of the U1 RNA was complementary to a sequence that was emerging as a consensus sequence for 5' splice sites. So we got very excited thinking that maybe the U1 SNRP would identify 5' splice sites and therefore be part of the cellular splicing apparatus. You can also use the antibodies to look at where something is inside cells. On the left are anti-ribosome antibodies marking the cytoplasm and the nucleoli within the nucleolus, whereas on the right is a picture of anti-RNP antibodies, which interact with the U1 SNRP alone, and you see that it's in the nucleoplasm, which is where the chromatin is being transcribed into RNA, and the RNA is being spliced from pre-messenger RNA to messenger RNA, so that fit with our idea. So we got very excited about this hypothesis. Let me first show you a picture of the people in the lab at that time. The person second to left from the left is Michael Lerner, who now works in biotech in California, a number of other graduate students and postdocs, and a colleague from Yale in this picture. However, not everybody else was as convinced as we were that this was the right answer to how splicing was carried out. And this commentary appeared in Science a couple years after we first published some of these ideas. And you can see the very last sentence there is the gloss is off the small nuclear RNAs temporarily, at least, as a solution to the splicing problem. However, we didn't give up, nor did various other labs, and very soon thereafter, two labs, or three labs, more than three labs, Phil Sharp's lab, Tom Manias's lab, Walter Keller's lab using mammalian systems, and John Abelson's lab using yeast, came up with in vitro splicing systems that actually carried out the splicing reaction, the removal of introns, which, as you see here, involves the attack of an intronic branch site A residue on the 5' splice site to give this lariat type structure that you see closer to the bottom of the screen. And then in the second step, the 2 exons are joined together. What was also realized was that this whole series of reactions required ATP, not because it goes into the intermediates or the products of the reaction, but because it's needed for many different helicases to be active as one goes from one state to another during this complicated process. And a lot of molecules move around and change their conformations. Another thing that happened, not in my lab, but other labs, is Alan Weiner's lab at Yale and Christine Guthrie's lab at UCSF did very elegant genetic suppression experiments where they made pre-messenger RNAs that had mutations in them that interfered with the splicing process, and then engineered into the U1 and the U2 RNAs, as shown here, compensatory mutations that would restore the base pairing. And indeed, they restored the splicing, providing very, very strong evidence that base pairing was involved between the RNAs in the SNRPs and the pre-messenger RNA in order to get splicing to go. So by the late 1980s, all of the 5 SNRPs you see here had been implicated in splicing. Moreover, each of them has a very highly conserved region within the sequence that biochemical experiments were identifying as being near the active site of the spliceosome, giving rise to the idea that we might really be looking at RNA catalysis, but protein assisted by the many proteins in the spliceosome. So also in other labs, this is from Ann Byer's lab at Virginia, very beautiful pictures emerged showing DNA with RNA strands coming off, particles building up at what was known to be the 5' and 3' splice sites. Those particles coalescing to form the spliceosome that you see labeled near the bottom of the slide with the intron looped out, about to undergo the second step of splicing and make the whole messenger RNA shorter. This slide fast forwards about 30 years and encapsulates the great excitement that started in 2015 with the first cryo-EM high-resolution images of the spliceosome. As you see in the top left, there are many, many states of the spliceosome, which is why X-ray crystallography could never get enough of one particular state in order to make crystals. But cryo-EM can overcome that problem and now work primarily from the 3 labs that you see listed in the top right, Yigang Shi's lab, Reinhard Lerman's lab, and Kiyoshi Nagai's lab, have established all these different states of the spliceosome, which makes very clear that what we really are talking about is RNA catalysis. And a lot of proteins, which you see move from place to place during different states of the spliceosome, the lower layer. So at this point, okay, I'm going to move on and talk just very briefly about alternative splicing, which is where not all the exons are included in the messenger RNA, and a single pre-messenger RNA can therefore give rise to several different protein products. About 95% of human genes are alternatively spliced. And what's also quite interesting is that splicing mutations now account for a greater and greater fraction of genetic diseases, perhaps up to 60%. But what I want to tell you about at this point, and the reason I'm introducing it with this alternative splicing side, is that there's one disease where knowing about splicing and alternative splicing has had a huge impact on patients. And that is the disease called spinal muscular atrophy, which is one where the the motor neurons of the spinal cord degenerate and become paralyzed at a very early stage in children. And this is most common genetic cause of childhood mortality worldwide. You see the numbers there. What was back starting in 1955, Judith Melke in France cloned the disease gene for this, which is called SMN, down here at the bottom for survival of motor neurons, and found that there were actually two loci on chromosome 5 because there's been an internal deletion or an internal duplication during evolution, and that it is loss of just one of these two copies of the gene that leads to the disease, despite the second copy, which is only 11 nucleotides over the entire gene, different from the first gene. With the result is rather devastating for the children who have this fault, as you see below. And at the very bottom, it points out that what the protein is a SNRP assembly factor, again, highlighting how important SNRPs are, and SNRP assembly, therefore, would be to the life of all cells of a higher eukaryotic organism. What was done, because of the fact that it's mutations of one of the two SMN genes, but there's another copy there that doesn't seem to be active, what was done was to study the copy that isn't active, which actually just simply isn't doing the right splicing, the right alternative splicing, and investigate the region around that part of the gene called exon 7 and all the different, and therefore could interfere with or enhance how splicing is done. And that's what this sort of frightening picture is at the top, and what the labs of the Singh lab, which was at the UMass Medical School and then at Iowa State, and the Craner lab at Cold Spring Harbor then did, was to make antisense oligonucleotides, short oligonucleotides about 20 long, that are complementary to various portions of the sequence, and just tile the whole region looking for oligonucleotides that would in fact relieve the inhibition and get exon 7 to be included, and therefore give a good product of the SMN protein, just like the other SMN gene does. And this succeeded, and subsequently has been turned into a therapeutic, which is called nusinersen or Spinraza. And here's a little girl who, as a baby, had lost the ability to sit, to stand, or even to sit, and has now been treated, and at the age of 3, in this picture, is doing quite well. And the text describes how the phase 3 clinical trials were terminated early because the drug had been so very successful. About a year ago, I heard from Adrian Craner that nearly 10,000 patients had been treated with this so far. There were no bad consequences, and nearly all of them had dramatically improved. So this is a wonderful story about how finding out about the basic research can lead you to therapeutics. Okay, so what I've told you about basically so far is how we started out with patients that had autoantibodies against various RNA protein complexes in cells and learned lots, and even with medical impact. And along the way, we had a lot of fun. Here are some characters that appeared at a Halloween party, and each one is a different SNRP with exons and introns in between them, poly A tail, etc. So what I'd like to do now at the very end is go ahead and say a little bit, as I promised, about mentoring in women in science. The next slide is basically advice for people who've been in my lab, whether they're male or female, about what you should do if you want to become a scientist, and just outlines that, if this is something that really gives you joy, you should go for it, and you'll figure out how to make it work. You need to expose yourself to a lot of kinds of diverse science, go to meetings, et cetera, take on the most challenging questions, and give people their own projects, because that way they own them and they get very excited about them and very interested in them. Stick to it, even when public opinion is against you, as it was for us in the early days of SNRPs and splicing. Diverse groups of people clearly come up with more creative solutions to problems. Some things about, taking vacations, choosing a supportive partner, which is really, really important, and I hear was extremely lucky. And do some teaching, because it expands your horizon. The next slide, encapsulates the problem for women in science. This is a slide that's pretty old. It also is really talking about the German academic system rather than the US academic system, but you can just substitute the names and you come up with the same sort of graph for the US, where about the same number of men and women in the biomedical sciences get PhDs, but then as one proceeds up the ladder to professor, at each stage, a man, the blue line, has a more probable outcome of being able to proceed up the ladder than a woman. In the US these days, we're doing a little better than the scissors graph would indicate, because we're at about in the upper 20% of women in the biomedical science, women professors in the biomedical science. So what's the cause of this, and what do we do about it? One thing that I was privileged to do was I was asked in 2005 to join a committee that was headed by Donna Shalala, who had been the HHS secretary under Clinton. And to write a report that mostly was about unconscious bias, which is a huge reason that we see a graph like this showed the one in the last slide. On the other hand, I also, shortly after this report was actually issued, and it's a wonderful report that I recommend to everybody, and there's implicit bias at the top, I learned about another phenomenon called social identity threat or stereotype threat that I think is equally important in terms of why women have not advanced so well in academic science. And what this phenomenon is, it's another phenomenon categorized by the cognitive scientists. It's when a person feels that they are part of a devalued minority. Certain physiological and cognitive reactions set in over which one has no control to create a state called vigilance. And when you think about it, since women in science are always in the minority, we're always in a state of vigilance, which does not help us either cognitively nor physiologically. And when I learned about this phenomenon, I was actually amazed, because I realized that for years I'd been sitting on committees where I was the only woman and the rest were men, and I felt like I really didn't want to say anything. And it was because I was undergoing stereotype threat. So my feeling at this point is I try to tell people about this, because just knowing about it and knowing why you're experiencing the reactions you're experiencing to various situations, I think helps one try to overcome them. And there's even evidence, as it says down here, stereotype threat prevents conceptual learning. Another thing I've noticed by mentoring many women, many men over the years, is that women tend much more to worry about the far distant future. My advice is to look ahead to the next step. Can you do the next step and not worry about whether you can do something that's 20 or 30 years away? So with that, I would like to simply thank all the wonderful people I've had in my lab. It's almost as much fun sharing the joy of discovery with a younger mentee as it is making a discovery yourself. And also my colleagues at Yale, my colleagues in the Yale Medical School and the international RNA community, and just a mention of some of my funding sources. And I just want to end by saying that for me, having had, he's now deceased, a very, very wonderful and supportive husband, Tom Steitz, was absolutely invaluable. I also owe support to that little guy there who just had his 40th birthday, my son John, and there we happened to be at Fred Sanger's second Nobel Prize party, where he won the prize for DNA sequencing rather than protein sequencing, the first Nobel Prize. So I'll leave it at that, and thank you very much for your attention.
Welcome to the 2020 ASN Business Meeting. We have 4 agenda items that we would cover at this meeting. We'll call the business meeting to order, recognize outgoing ASN leaders, and pass the gavel of authority. Dr. Mark Rosenberg, past president, will present the report of the 2020 ASN Nominating Committee. President-elect Sue Quagan will present the outgoing President's Medal and celebrate the 2020 recipients of the ASN's awards. And finally, Secretary-Treasurer Dr. Keisha Gibson will update the status of ASN's finances. So I'd like to call the 2020 ASN Business Meeting to order and recognize our outgoing ASN leaders and pass the gavel of authority. So the outgoing ASN leaders to be recognized include Dr. Ziad Al-Ali, who served as the chair of the ASN Publications Committee, Dr. Michelle Josephson, who served as the chair of the ASN Policy and Advocacy Committee, Dr. Kambiz Kalantari, who served as the chair of the ASN Career Advancement Committee, Dr. Richard Lafayette, who served as the editor-in-chief of ASN Kidney News, Dr. Pascal Lane, who served as the chair of the ASN Media and Communications Committee, Dr. Mark Parazella, who served as the education co-director for our ASN board review course and update, Dr. Paul Sanders, who served as the chair of the ASN Grants Review Committee. I would like to present the ASN past president's plaque to Dr. Mark Rosenberg for his outstanding service and leadership to ASN, serving on council for 7 years. I'd like to now pass on the gavel of authority to incoming president, Dr. Sue Quagan.
I would like to present the report of the 2020 Nomination Committee. I had the honor to serve as chair of this committee, and the members of the committee you can see here that include Michelle Konkel, Tamara Isakova, Matthias Kreitzler, Karen True, and Suzanne Watnick. The result of the election is shown here. ASN president will be Susan Quagan, president-elect Barbara Murphy, and the 2 newly elected councilors are Michelle Josephson as the secretary, and the at-large councilor is Patrick Nachman. With the results of this election, you can see on this slide the 2021 ASN Council. Thank you.
Hello, I'm Sue Quagan. I'm president-elect of the American Society of Nephrology, and I'm absolutely delighted to present the outgoing ASN President's Medal and to celebrate the 2020 recipients of ASN's awards. This year's outgoing ASN President's Medal is awarded to a colleague, friend, and fearless leader, Dr. Anupam Agarwal. He will become past president in 2021 to serve his 7th year. To say that it has been an unusual year to be ASN president is an understatement. I can't thank Dr. Agarwal enough for all he has done for us on council and for all the members of the American Society of Nephrology. This year's 2020 recipients of ASN's awards are the following: Sophia Ahmed, Distinguished Mentor Award; Nisha Bansal, Distinguished Researcher Award; Joseph Koresh, Belding H. Scribner Award; Giselle Duder, ASN President's Medal recipient; Derek Fine, Distinguished Clinical Service Award; Anna Greca, Donald W. Selden Young Investigator Award; Leslie Inker, Distinguished Researcher Award; Shuda Ashibe, Distinguished Researcher Award; Holly Kramer, Distinguished Leader Award; Tu Lee, Distinguished Mentor Award; Kathleen Liu, Distinguished Leader Award; Vandana Dua Niyar, Distinguished Clinical Service Award; Priti Patel, ASN President's Medal; Professor Sir Peter Radcliffe, Homer W. Smith Award; John Sperati, Distinguished Educator Award; Ravi Tadani, John P. Peters Award; and Ashita Talwani, Robert G. Nairns Award. Please join me in congratulating all of these recipients.
Good morning, everyone. It is my pleasure and honor to provide this year's Treasury report. ASN's financial portfolio is divided into our operating reserve funds and then our 501c3 foundation, Kidney Cure. As you will remember, Kidney Cure was established as an outgrowth of ASN to provide a mechanism for private individual and corporate donations to be directed specifically towards research, and to create endowments to make sure our grant programs will survive and be funded in perpetuity. Kidney Cure funds our fellow grants, career awards, and other awards. In addition to supporting salaries and other operational costs, 4.5% of our operating funds are used for special projects like Kidney X. You may note on this slide that ASN's operating budget has seen a slight decline over the last 5 years. But as you will see, this is a function of higher expenses that include things like funding Kidney X. Our overall trend in revenue continues to increase, and altogether we've seen a 5-year growth rate of approximately 6.3%. The largest source of our revenue includes educational activities like Kidney Week. Over the last 5 years, the portion of our revenue represented by educational activities has remained fairly stable, ranging from 57 to 61%. Another 30% of our revenue is represented by communications like our journals and membership dues. Together, education and our communications represent a little over half of our expenses. This is a bit of a decrease compared to 5 years ago, where together those entities represented about 3/4 of our expenses. The other category that you'll note in this slide represents other administrative costs. Together, our ASN and Kidney Cure investment portfolios have performed well, and from year 2018 to 2019 grew from about $90 million to $115 million. Total assets for ASN and Kidney Cure over the last 5 years have grown from about 75 million to now close to 120 million. Finally, our liabilities have ranged from just under $9 million to just above $10.5 million over the last 5 years. In 2019, you will see here that we landed just about in the middle range of this range. Part of this decline that we've seen is due to the recent sale of our previous condominium office space and the signing of a new lease agreement. So overall, I'm very pleased to report that our financial status in ASN remains strong, and we are looking forward to seeing all the things that our organization will continue to be able to support. Thank you for your attention.